to Spotlight on Antibodies, a podcast from BioIntron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in June, recent research includes VHH antibodies, antibody-drug conjugates in oncology, and antibody engineering as a countermeasure to drug resistance. First up, an introduction to VHH antibodies, also known as single-domain antibodies or nanobodies. These are the smallest functional fragments of antibodies, consisting of a single variable domain derived from heavy-chain-only antibodies found in camelids or engineered from human immunoglobulin frameworks. Despite their small size, they retain full antigen binding ability, and their high solubility, stability, and ease of genetic manipulation have positioned them as powerful tools in diagnostics, therapeutics, and biotechnology. Recently, researchers developed a modular synthetic nanobody phage display vector that allows for precise manipulation of binding and framework regions. This platform overcomes key limitations of camelid immunization, enabling library generation for non-immunogenic or toxic antigens, and facilitating pharmacokinetic tailoring. The resulting library yielded a nanobody cross-reactive with human and mouse calreticulin and suitable for PET imaging when radiolabeled. These findings established synthetic libraries as a scalable foundation for generating clinically relevant single-domain antibodies, supporting downstream efforts in solubility and delivery engineering. Further downstream, a recent study focused on improving the solubility of single-domain antibodies for therapeutic development. They examined two VHH antibodies with human VH-like hydrophobic hallmark residues that exhibited low solubility despite being derived from an alpaca immune library. By introducing VHH-like mutations into framework region 2, they successfully increased the solubility of one VHH antibody without buffer additives. For the second VHH antibody, additional modification was required to achieve monomeric behavior. Both engineered VHH antibodies retained functional integrity, demonstrating that solubility can be rationally improved even in the absence of structural information. Besides solubility, researchers are advancing the pharmacokinetics of VHH antibodies. A recent study introduced a universal site-selective conjugation strategy for variable domains using thiolmaleimide chemistry. This platform supports robust and customizable bioconjugation without compromising antigen binding activity, facilitating the development of VHH antibody-based diagnostics and therapeutics. Such conjugation methods are particularly advantageous when integrated with delivery strategies targeting mucosal or systemic compartments. On the treatment delivery aspect, recent work has demonstrated the potential for topical or oral delivery. A recent paper describes human VHH antibodies for non-invasive, localized topical treatment of mucosal inflammatory diseases. In a murine study, the anti-IL-33 VHH antibody, named A12, was delivered topically and via inhalation to target inflammation at the ocular surface and in the lungs. A12 effectively inhibited IL-33 signaling, reduced dry eye severity, and ameliorated allergic asthma symptoms. These findings highlight the potential of VHH antibodies for targeted modulation of mucosal inflammation through localized delivery routes. Meanwhile, oral biologic therapy has been a long-standing challenge due to protease degradation and poor gastrointestinal stability of conventional antibodies. Researchers engineered an anti-interleukin 23 receptor VHH with both high affinity and protease resistance, achieving picomolar binding through contributions from both CDR and framework residues. The optimized VHH remained stable for over 8 hours in intestinal fluid and 24 hours in fecal samples. Oral administration in mouse and cynomolgus monkey models led to deep, sustained inhibition of IL-23R signaling, validating functional delivery to the gut. These findings establish orally delivered VHHs as a promising therapeutic platform for chronic inflammatory diseases, such as inflammatory bowel disease. Last month, another study reported on a panel of VHH antibodies targeting the conserved S2 subunit of the SARS-CoV-2 spike protein, offering broad and potent neutralization of both SARS-CoV-1 and SARS-CoV-2. One VHH in particular bound a highly conserved quaternary epitope locking the spike protein in its pre-fusion conformation. When fused to a human IgG1 Fc and administered systemically at low doses, this VHH prevented SARS-CoV-2 infection in 2 animal models. This work highlights the therapeutic potential of SDABs as next-generation antivirals and underscores their unique ability to target structurally conserved epitopes inaccessible to conventional antibodies. Besides VHH antibodies, another format gaining popularity is the antibody-drug conjugate. Antibody-drug conjugates, ADCs, couple the specificity of monoclonal antibodies with the cytotoxic potency of chemotherapeutic agents. Recent advances have been reported in both hematologic and solid tumors, with next-generation ADCs overcoming limitations of traditional therapies through improved targeting, linker technologies, and payload selection. Recently, a new ADC was reported showing promising safety and response rates for patients with a rare blood cancer, blastic plasmacytoid dendritic cell neoplasm, or BPDCN. According to data from a phase 1 and 2 study, pivekimab sunirine, or PVEK, achieved an overall response rate of 85% and complete response rate of 70% as frontline treatment in newly diagnosed patients with BPDCN. PVEK is an ADC directed against CD123, a subunit consistently overexpressed in BPDCN. With a high-affinity anti-CD123 monoclonal antibody conjugated to a potent cytotoxic payload via a cleavable linker, PVEK enables targeted intracellular delivery of its drug component following receptor-mediated endocytosis. Compared to earlier agents, which used a fusion toxin approach, this ADC format allows for improved pharmacokinetics and potentially broader therapeutic window. Meanwhile, DB1310, a HER3-targeted ADC, exhibited promising early clinical activity in patients with advanced solid tumors resistant to standard therapies, particularly in those with EGFR mutant non-small cell lung cancer. DB1310 is a novel ADC comprised of a humanized anti-HER3 IgG1 monoclonal antibody, cleavable peptide linker, and DNA topoisomerase 1 inhibitor. Further along in the clinical research space, new phase 3 data has positioned Trodelvy, a TROP2-targeted ADC, as a potential new standard of care in PD-L1 positive metastatic triple-negative breast cancer. Trodelvy delivers the cytotoxic payload SN38 to cancer cells, where it inhibits topoisomerase 1 and induces cell death by disrupting DNA replication. In a competitive ADC landscape, Trodelvy faces emerging challenges from agents such as Kelun-Biotech's sacituzumab tiromotecan and AstraZeneca's Datroway. Additionally, China has approved trastuzumab deruxtecan, a novel HER2-targeted ADC, for adult patients with unresectable, locally advanced, or metastatic non-small cell lung cancer, harboring activating HER2 mutations who have received prior systemic therapy. Beyond its established efficacy in lung cancer, this ADC has demonstrated clinical advances across multiple tumor types with 8 additional indications. Finally, let's move on to the topic of antibody engineering as a countermeasure to drug resistance. Resistance treatment continues to limit the long-term success of many therapies. In oncology, cancers may evade antibody-based treatments through mechanisms such as antigen loss, impaired intracellular trafficking, or suppression of immune responses. To address these challenges, researchers are developing advanced antibody formats, including dual payload antibody drug conjugates and multi-target T-cell engagers, which are designed to maintain efficacy even in the face of resistance. These innovations combine different modes of action to enhance therapeutic durability and reduce the likelihood of escape. Meanwhile, monoclonal antibodies are being applied to the detection of antibiotic resistance, such as beta-lactamase enzymes, offering new tools for rapid diagnosis and targeted intervention. Together, these efforts reflect a growing focus on using antibody technologies not only for direct therapeutic action, but also to anticipate and overcome resistance across diverse clinical settings. In response to the persistent problem of therapeutic resistance driven by tumor heterogeneity, recent research has focused on dual payload ADCs that integrate different cytotoxic mechanisms within a single platform. These novel constructs combine chemotherapeutic agents and radiotherapeutic isotopes to exploit synergistic interactions and enhance anti-tumor efficacy where single payload ADCs fall short. The inclusion of radiometals paired with platinum-based DNA-damaging agents can enable increased cytotoxic reach via bystander effects. In parallel, radiolabeled ADCs offer dual utility for both therapeutic and diagnostic applications, as their imageable isotopes facilitate tumor visualization. Design parameters such as the type of radioactive decay, linker chemistry, and drug-to-antibody ratio are being optimized to maximize therapeutic index while minimizing off-target effects. Although none of these constructs have yet to advance to clinical trials, preclinical data and human equivalent dose modeling suggest favorable safety profiles and potent efficacy. Despite the clinical success of several FDA-approved ADCs, almost all patients develop resistance via multiple mechanisms. The resistance arises through various mechanisms such as alterations in the target antigen, decreased internalization of the ADC antigen complex, lysosomal dysfunction impeding payload release, and changes in tumor cell sensitivity to the cytotoxic payload. The choice of linker further influences ADC performance, particularly in relation to the bystander effect. Cleavable linkers facilitate payload diffusion to neighboring cells, enhancing cytotoxic reach, while non-cleavable linkers offer greater serum stability but reduced bystander activity. Additionally, the drug-antibody ratio must be optimized to balance therapeutic potency with systemic toxicity. Strategies to overcome resistance include rational design of target antigens, selection of more effective linkers and payloads, and consideration of the immune effector functions conferred by IgG1 isotypes. Bispecific T-cell redirecting antibodies have also demonstrated significant efficacy in multiple myeloma. However, both primary and acquired resistance limit their clinical benefit. Resistance mechanisms are multifactorial and include tumor-related factors such as high tumor burden, antigen loss, and expression of T-cell inhibitory ligands. Primary resistance often correlates with impaired baseline T-cell function, while prolonged exposure can lead to chronic T-cell stimulation and exhaustion. Combinatorial strategies such as using CD38 targeting antibodies or developing trispecific antibodies to counteract antigen escape are currently being investigated to enhance T-cell activation and improve response durability. Understanding these resistant pathways is essential to optimizing the clinical utility of bispecific antibodies in multiple myeloma. To address the urgent need for rapid diagnostics in the face of rising antibiotic resistance, researchers have also developed monoclonal antibodies targeting a conserved epitope within AMP-C beta-lactamases, which are enzymes central to resistance against beta-lactam antibiotics. These monoclonals offer a novel tool for direct protein-level detection of beta-lactamase activity. Their integration into diagnostic workflows could improve surveillance and guide appropriate antibiotic use through clinical, veterinary, and environmental applications. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.